translate it, and I try to answer. Or do you prefer German?
No.
No?
In English.
English. Okay.
Yeah.
Good. So, what I want to tell you, because we know each other for more than 10 years, Hilda was doing her PhD at Munich in my lab with that, and mainly with atomic force microscopy. I moved, and now I'm heading a research group for radiation, biophysics. And you are all aware during your physics studies what radiation is. The interesting thing is, when you reflect this, yeah, a total dose of 4 gray, you know the abbreviation of the dose gray, yeah, you know. 4 gray, one shot of 4 gray can kill a person. But what are 4 gray? Because you're physicists, you know how much energy is in there, how many joules. When you now think on that, when we irradiate now our body with 4 gray, this is around, and we take a normal person, and the normal person has 70 kilograms, and we irradiate him with one shot of 4 gray. These are only 67 calories. And these 67 calories can kill the person.
What?
And what are now 67 calories? If you translate this, one sip of coffee. It's the energy of one sip of coffee. This energy can kill your body of ionizing radiation. Or, if you're strong enough, you're lifting a person for 0.4 meter. This is the energy that can kill a body according to ionizing radiation. Forget everything with the atomic bombs. Yeah? Much more energy. So, and what is now happening in terms of biophysics? In terms of biophysics, it looks like that. Here are our cells. Here we have the ionizing radiation. And when we have ionizing radiation, you know we have alpha particles. These are particles. And you can easily shield with one piece of paper. Put the paper on top, the radiation is gone. Beta particle, take a piece of plastic, gone. But X-ray and gamma ray you use for medical examinations can pass the body. And what is now happening with this energy passing through the body? It now, in the first part, it ionizes the water. It's the so-called hydrolysis of water. In billion parts of seconds, radicals are formed, and these radicals now interact with your particles inside the cell, with DNA, with proteins, and so on. Okay?
Okay.
Now, next step is that it can lead to a DNA damage. I will show you after how often this will happen, even when we are sitting here. Because we are right now also exposed to ionizing radiation, depending from the soil. It's a very big amount. I can have a look further. Brazil, one beach in Brazil, I think in the Bahia area, is a monazite. It's a mineral, crystal beach. It's a black beach. Probably heard about that. The same is in India. The radiation in this field is 200 times higher than on the other beaches. Yeah?
Oh.
It's on that. I can look for the numbers if you want. Yeah? Okay, so DNA is one target. DNA will be repaired, then it will, no problem. When there is not a 100% repair, this will cause now effects. All you know, it can cause a tumor, cancer, and in the end death. So, therefore we have to know what is happening there, how can we overcome these problems, and how can we, in terms, can we reduce radiation, what's necessary. Of course, we cannot reduce the radiation from soil. This we cannot do. But we can reduce the X-ray images. Every time when you go to the doctor, he says, okay, let's come, take an X-ray. Take an X-ray. And in my group, we are interested how many X-ray, so to say, are necessary, or they cause harm. On one hand side, you image every time, yeah, okay, now now your arm is fine, but unfortunately you have now skin cancer. Yeah? So you have to find the balance. What is necessary? And this is also some kind of radiation protection. Okay? So what we have, this is now one sketch what we developed together with our collaborating partners in California from Berkeley. This is now the effect, what we know from high doses. High doses are mainly when you're exposed to, let's say, you are during an operation, chemotherapy, atomic bomb, when you're in the center. What will happen? And look just on the time scale. In parts of seconds, it's now the physics when ionizing radiation is coming, will hit the molecule, and of course when radiation is coming, you know microwaves, you know infrared, yeah, or when you're in the sun. Heat. Then the other part is ionizing of the molecules, water. These water radicals now react with all your DNA, protein molecules, membranes. Proteins and membranes are replaced in the next cell division. But if DNA is damaged and is not 100% repaired, this will cause a heredity problem. And this will mean a tumor, cancer, or death. Or you transport this so-called DNA mutation to the next generation. So therefore it's very necessary that we understand possible effects there. This DNA damage can take minutes to hours, but then now the big problem starts. All the rest effects can take days or even decades, up to 12 to 20 years. So normally, when you do an experiment, or when I take a knife, I cut myself, I see directly the effect. It's bleeding. When we now are talking, yeah, and a radiation source, for example, is passing by, we are irradiated for 1 second. You will not feel it. And in 10 years later, you get a cancer. Can you judge, okay, it was according this event or not? And this is quite a problem. Now you see the problem where we are in now. This is now the effect of high doses. Now when we go now to this step, what one X-ray image is doing, we have to add a little bit more, and this we call the cell microenvironment. As you know from biology, cells are communicating to each other, not with nerves, but also via cellular gaps, gap junction, calcium flux, and so on. And this is part of my research, that we control this, that we have now biophysical tools, we develop biophysical tools that we can follow this. Okay? Good. This is now actually the entire discussion what we are facing. But I want to show you just a few spots that I can show you a little bit more on the stuff what we are doing. So, we have our ionizing radiation. Then we have specific factors. What are the effects? It's the same when you go into the sun. When I go into the sun, after 5 minutes my skin is burned. If you go into the sun, you probably 1 hour. Because your body is adapting. You can adapt persons to radiation in specific amounts. One. Then, when you compare yourself, you can probably stay 1 hour, 3 hours in the sun. So, your genetic susceptibility, your genes are also important. So, even one additional factor that is very difficult that we can say, okay, this dose corresponds to this cancer. Next problem. So, and to sum this entirely up, the theoretical guys in radiation sciences developed this model, LNT, linear dose response model. What this means is the following. Here we have the dose, and here we have effect, any effect, cancer or whatever. Now, these are math guys. Yeah? What they have is what we know. We know effects with high doses. Atomic bomb survivors in Japan. Hiroshima, Nagasaki. Okay, we know we have a high dose with high effects. Okay, and now when we're thinking on Mars, when we do not Atomic Cafe. You will love. Atomic Cafe. If you don't know this, excellent. They made, how do we call this, contest? This universe. Beauty contest. They made beauty contest for the election of Miss Atomic Bomb. The ladies posing in bikini behind the atomic bomb. No joke. Look at the movie. Okay. Now, how are these information coming from? So, from direct studies from the low and high dose phase. As I mentioned, Hiroshima and Nagasaki. What you see in this graph are, depending on the color, the ionizing dose they achieve the person, and this is exactly one of the most expensive studies ever. When you look on this medicine, each dot corresponds to one person surviving the atomic bomb in Hiroshima. It was done in the Leavenhauer Institute on that. So here was the epicenter, and then you see how the wind was going and the ionizing radiation cloud was transported. Then, if you have this one, statistical significant differences you cannot expect after so many years. And what is your normal background ratio? Genetic, adaptive response. For example, when you have now cancer, is this cancer due to the ionizing radiation? Or, for example, when you are smoking, you add the risk of lung cancer dramatically. And if you're exposed to ionizing radiation, the factor goes up for 20%. Thank you. So the other thing is, you have to know what you are doing in terms of radiation. And this is one of the work stores in America where they used to paint watches. Probably you notice when your watch is going to the dark, your numbers are fluorescent. What they were using a fluorescent dye which was radioactive, the painters. How do you do when you paint something small? You take the stick, paint like that, right? And they used it with a paint, radioactive paint, every time. When you study this, there is a study on that, on these dial painters. It can directly be proven that all the chest and, let's say, face tumors that occurred are corresponding to the work what they did here. So, be careful on that what you're doing. Okay, now, back to that. Okay, this we know, so therefore it's very hard to distinguish positive and negative effect and so on. Yeah? And this is now exactly where biophysics starts. What we have now, we can have a qualitative, like mechanics studies. When I irradiate cells, I check the consequence on the cells. I see the DNA changes, I see the DNA destruction, but what does this mean? If you have 10% DNA destruction, 5% repair possibilities, what will this mean in the end? What will the doctor use for that? Will he say, okay, 10% cancerous, 5% cancerous? This is the problem. So, what we are looking for are so-called biological endpoints. When your skin is burned in the sun, this is a biological endpoint. You definitely know, don't go into the sun. On the other hand, quantitative, epidemiological studies. If you are aware now, a few months ago, starting this year, we had this Fukushima incident in Japan. I can show you the data of what we could record in Munich, according to the radiation fallout. So it's necessary that we know this. And we in Europe, nearby, probably you know this from history, 1987, there was the incident in Chernobyl. Chernobyl, this was where the atomic power plant in the former Soviet Union exploded, and a massive fallout also passed Bavaria, Germany. Up to now, we are not allowed to eat mushrooms from the Bavarian forest. It is still contaminated. 20 years ago. The radiation cloud is gone, but we have the fallout of the nucleotides. Cesium 2,000 years. Even in the next 10,000 years, we have still this radiation. The guys in Japan have this for also the next 10,000 up to 100,000 years. And this is what we have to know. Even when we shut down power plants, we still have the waste. Or we have to put the waste. I do not know where the Brazilians put their waste from their atomic power plants. The Germans also do not have an idea. I think worldwide there is no one working approach to store atomic waste from the power plants. You cannot put it into the sea. It will come back. Not in this generation, but in the next generation. Yeah? Okay. And now, to cross all this, and to bring a little bit more biophysics in there to clear this process, in my lab, what we develop lab-on-a-chip systems. I will show you. We have an operating lab-on-a-chip system, you know probably from your lectures. Lab-on-a-chip is the chemistry laboratory in the size of a fingernail. I do the entire biochemistry in this size. A single cell detection, for example. I show you. There's a lot of physics in there. This I show you. Then we developed sensors, semiconductor sensors, that are able to record online the ionizing radiation from a low to a very high range. Then we have, together with the University Unicam in Campinas, we are developing lateral flow immunoassays to detect now diseases on them, and then we let you know we have 2D manipulation, and then we now expanded this to 3D manipulation. We have now AFM grippers with a haptic control that we can really feel when we move something. Okay, it's like a toolkit, a platform. Okay, keep this in mind. These are our cells. Proteins are altered after radiation, double strand breaks of the DNA. Therefore, we have our system, and I will get through a few ones. As I promised before, every time we are exposed to ionizing radiation, what you achieve per year are 4 millisievert. You cannot do anything against it. 4 millisievert you will get definitely. 2 millisievert are from natural, depending where you live. In Bahia at this dark beach, probably more. And 2 millisievert you get from medical examinations. So, when you now think on radiation protection, next time you go to the dentist or to the doctor, ask him if this is really necessary, and ask him, please ask him, what is the dose? He will probably use his old-fashioned instrument from Röntgen. They're using a very high energy. It's your health. Yeah? So, for example, when you get 1 gray, 4 gray are lethal, then you're dead. 1 gray radiation causes in our body 2,500 base damages in the DNA, 1,000 single-strand breaks, up to 40 double-strand breaks, and up to 80 cluster base damages. Quite a huge number. But the good thing is more than 99% can be repaired. Otherwise, we would not sit here. Okay? So, now gets to the first part, to this lab-on-a-chip system. When you're talking on lab-on-a-chip systems, you have to face in terms of physics a lot of problems, because you shrink down your normal lab into the size of a fingernail. One problem. The other problem is when you consider this as a tissue, normal tissue or what you take from the pathologist, how can you get one single cell out of this tissue, precisely one cell, and position it very precisely? And this is what I want to show you right now. And this is a little bit inspired from biology. By training and biology. Okay. When you see this hummingbird, how he's doing this, he's sucking. Yeah? Okay, this we do. And here on the spider net, you see all the properties. Now think as a physicist. How you can handle and manipulate droplets. Just don't think as a biologist. Think as a physicist there. It's a hydrophilic, hydrophobic interaction. Right? Mm-hmm. Depending on the diameter of these spider net fibers, smaller or bigger droplets can adhere. So it's only a chemistry blocker. Hydrophilic, hydrophobic interaction. Okay. And this is how we developed in the lab. We have now such a hummingbird trunk which can absorb single cells after manipulation with a laser. I show you. And this is now our chip system. What I'm going to show you. These are two heating devices. One, the second. Here we can perform polymerase chain reaction. You came across it in your studies to amplify DNA. So we can do here, and this small red droplet, I stained it, is 1 microliter. We do all our experiments in 1 microliter, not more. And now to avoid this evaporation, yeah, we seal it with mineral oil. That's all. Okay. Let's go back. How can we get access to the cells? And this is the topic of microdissection. It started in the Middle Ages. So when the doctors came here, they took knives and cut up. Okay. In the 70s, in medicine, they used extended glass needles. They took a glass capillary, heated up, and pulled. And then in the end, you had a diameter of these pipettes around 1 micrometer. Then, after the invention of AFM and all this stuff, we were using the AFM cantilever tip to microdissect DNA. This was actually the time when Lila went over also to Germany. And then we have another approach, but this is contact. The risk of contaminating the sample is very high, because we only want to work with 1 cell, not more. Then we developed also together with a big company, and is now also available on the market, as a principle where we use a laser. We use a UVA laser. UVA, when you check the spectrum of UVA lasers, it's far away from the absorption maximum from DNA and proteins. So we do not alter the molecules we are interested in when we manipulate. Okay? And how we do this is shown in this movie. So we take a normal glass slide, put a plastic membrane on top. On top of this membrane, we put our histological material. Then we take the laser, focus directly on this level, optical level, and circumscribe by moving the stage. It's like a laser sword in Star Wars. Yeah? And then what we do is, now we take this trunk and absorb this small particle and release it precisely. This is how it looks like in the lab. This is now an inverted microscopic stage. Here we have our sample. This is now the trunk device. This is a German bird's name, Spatz. You know the small one? Yeah. But this is abbreviation for single particle absorbing transfer system. Okay. Took me quite a while to get this name. Okay. Now the trunk is approaching. Here you see the isolation of a single nucleus. Here. This is the lattice grid inside the trunk. Absorbed. It's still in there, and then it's released precisely. Here's the chip system. And it's precisely released in this droplet. The big part of this system is only the cooling device for the PCR to run very fast. But you will see in a second. Okay, this is how it looks like. This is now a single nucleus, what you can isolate. And they are precisely released in this droplet. The volume of these droplets is 200 nanoliters. You can do it with cell clusters. You can do it with living cell material. So now, now you can isolate the cells. Now we have to process them. Processing them, the big problem in microfluidics, as I mentioned before, is when you shrink down this tubing system, laminar and turbulent flow is important. Normally, when you shrink down the diameter in a stick, for example, all flow is laminar. You have no mixing any longer. And the next part is, according to the increasing Reynolds number, viscosity is increasing. You have the same when you, for example, take a stick and soak in water. This you can do by lump. Try to do this with honey. No. This is the problem what you have in terms of microfluidics. how we overcome this in our lab, the first one is we do not have any three-dimensional structure. What we have is we have 1 microliter of reaction volume, maximum 5, so we take the surface tension of the droplet, nothing more. And to avoid evaporation, we seal with mineral oil. Then, to move the droplets in a given way, think on the spider net, we generate a hydrophobic tracking system on top of our chip, of our glass. This is done with lithography method. It just takes Silan chemistry. And for actuation, that we can move the droplets, we do not need a pump. What we do is we take the so-called surface acoustic waves. You know surface acoustic waves? Surface acoustic waves, everybody is carrying with you, but you don't know this. This is the damping device, the noise reduction in your mobile. Yeah? What we are using, it's an acoustic wave. And this acoustic wave is now getting into contact with the droplet. And then the droplet starts to move. This is a 50 nanoliter droplet, only moved by surface acoustic waves, no pumping. Here you see the pumping of the wave. Yeah? If you take a bigger droplet with the same energy, it's like a sausage you can move. And this corresponds now, let's say, to a laminar flow. Now, very important is the angle here, how this surface acoustic wave is getting in contact with the droplet. At a specific angle, the angle is called the so-called Rayleigh angle, is coupled into. Then we change this angle, make it a little bit larger, then we can switch from laminar flow inside the droplet to mixing. It's still the same droplet. We are mixing in the droplet. Okay? And now think back, how can we change now this angle? It's just the surface chemistry here. How the droplet sits on the glass. And this we can easily modify when we change the surface chemistry. This means only by changing the surface chemistry on our chip, we can let the particles move, can mix, and can process. Nothing else. Okay? Then we implemented in this system a little bit more, because you cannot pipette in 1 microliter precisely with a pipette. So we developed a dispensing system, mainly based on inkjet technology, what you have in the computer, yeah, with the accuracy of 10 nanoliters. Then we have a fluorescent control as a readout system, and everything is put together in the system how it looks now in the lab. Here's the chip system. So here we have the laser installed, here is the sample, here is the transfer trunk, moving over there. Then on the slider you can pass over there to this nanopipetting unit. Then you move and process the samples, and then you can have a fluorescence readout. And how it looks like is the movie shown here. This is how it works in the lab. Again, this you have seen. This is the membrane, here we isolate, and everything is done just by the joystick. The students are sitting there with the joystick. Here's the nucleus, isolated. Here you see the lattice of the grid. So in. Then you release it on top of the chip system. Here's the droplet, here's the mineral oil. And what you then can do is, then you fuse just with the surface acoustic waves. Everything is non-contact. Yeah? Then you come to the nanopipetting unit. Here we have all our biochemical reactions prepared. Just soak in. It's piezo driven. It's a piezo crystal pressing on the stick. Then we take this one, go down there, releasing precisely the biochemicals, 10 nanoliter accuracy. Then you can fuse Now you can perform PCR, for example. Then you can incorporate also a fluorescent dye. Here's a reader what we constructed. We illuminate the source and check the fluorescent signal in the droplet. And the more cycles we run, the more DNA is produced, the brighter gets the signal. When you once perform the PCR, normally in these Eppendorf tubes, PCR takes you 2.5, 3 hours. We do it in 20 minutes, according only to the small volume. Because the major part of all this biochemical reaction is heating up, cooling down, heating up, cooling down. And in 1 microliter, heat up and cool down very fast. Okay, good. Let's get back to our double-stranded DNA breaks. How can we detect something like that? Okay, we have single-strand breaks, double-strand breaks, but much of these are repaired. So there is now one immunochemical detection system that is operating with specific antibodies that can directly go to the areas where a double-strand break happens. A lot of research groups around the world are exclusively studying this because it's a highly complicated system. But we are not very much interested in this one. We are using these antibodies. Yeah? And what will happen with that is this one. So these are now nucleus where the DNA is inside, fluorescent image. When you do fluorescent imaging of DNA, you use a blue dye. The blue dye is dark heat. When you check the fluorescence, it's blue. 99.9% when you see a fluorescent image of biological material, and if something is blue in the center, this is the nucleus, this is DNA. Now, we take the antibody, this is directly going to the repair site where you have a double-strand break damage. You see here as a point. Yeah? And each point corresponds to a DNA repair foci. Now, depending on the dose you apply, the more points you get, and depending also on the repair time. How long do you allow the system to repair yourself? You see, here's the repair time increasing, and the foci decrease. Okay? And this is now what I mentioned before as a biological endpoint. In our lab, we did the following. What we had our droplet setup, now with 40 droplets. In each droplet, there are living cells. Then we shield everything with lead against ionizing radiation and irradiate it. So we have here, in the first line, 1 grade irradiation. You see the repair dots? 250 milligrade, less. Yeah? 100 milligrade, even less. So this is what students have to do in these studies. We are taking these experiments, taking the repair time, changing, let the system control yourself. And then we check how many DNA repair foci are prominent as an endpoint marker. Okay? And then you have an idea what is the impact of ionizing radiation to this tissue. Okay? Good. Now when we think back on the problem, can this system, what I showed you, detect cancer or alterations in the DNA? This is what we did here. This is a colon cancer, the polyp. It's a typical cancer when in a specific gene in the genome, one base pair from 3 billion is deleted, or up to 5. And this we thought, hey, this could be a very nice approach to prove the power of our system. So what we did, we had material from patients, we had blood samples, we isolated from the blood single lipocytes, nucleus, then we isolate this funny shape. You find it directly, this funny shape again in the droplet. Okay, it's the same. Then we performed a lot of PCR. I don't want to stress you with that. This takes you 1 hour. Yeah. After that, we took this material and put it to sequencing. Only one nucleus. Healthy deletion of 1 base pair, second patient, healthy 5 base pair deletion. then we were checking, not for this deletion, but it's everywhere, every time there, yeah? Okay, we were checking for proto-oncogenes, for genes that are activated before cancer. This is BRAF, KRAS, and also the stability of the chromosomes is very important, the microsatellite stability. And when we check now here for BRAF, you see a mosaic, for BRAF and KRAS, you see a mosaic, and for the microsatellite. It's not a homogeneous tissue. So, and now, this is, let's say, one of the first approaches to improve radiation therapies, but also cancer therapies. When you now have this information, you believe that this tumor is still a homogeneous one. Get one chemotherapy. It will probably kill only a few cells. This is what you have to know. And also with the radiation. This is not the question what we can solve, but we have the technology in terms of biophysics to do that. Yeah? This is the first problem. And the last minutes I show you the second problem. This was the actual cartoon provided by the Japanese government to the school kids and to students, explaining the Fukushima incident. It's also you can download